Hello friends, today in this lecture we will study about the different types of blastula. But first, before moving to the types, we will study about the basic events of embryogenesis. The process of embryogenesis begins when an egg or ovum is fertilized by a sperm cell to form a zygote. This zygote then undergoes mitotic division, a process that does not result in any significant growth but creates a multicellular cluster called a blastula. This process is referred to as cleavage. The rapid, multiple rounds of cell division are termed cleavage. Cleavage can take place in two ways: holoblastic, total cleavage, or meroblastic, partial cleavage. The type of cleavage depends on the amount of yolk in the eggs. In placental mammals, including humans, when nourishment is provided by the mother's body, eggs have a very small amount of yolk and undergo holoblastic cleavage. Other species such as birds with a lot of yolk in the egg to nourish the embryo during development undergo meroblastic cleavage. As the amount of yolk in the egg increases, it influences cleavage by hindering the cytoplasmic movements involved in mitosis. If there is only little yolk, oligolecytal eggs, the yolk granules follow the movements of the cytoplasm and are distributed in the resulting blastomeres. But if the amount of yolk is larger, megalecythal eggs, cleavages occur nearer the animal pole where there is less yolk. As a result, the blastomeres nearer the animal pole are smaller than those nearer the vegetal pole. The presence of yolk masses may retard the onset of cleavage in a part of the egg or even suppress it altogether. In this case, cleavage is partial or meroblastic. Only a part of the egg material then is subdivided into cells, the rest remaining as a mass that serves as nourishment for the developing embryo. Both holoblastic and meroblastic cleavages give rise to blastulas that can be classified into two types. First, in one type of blastula, all the cells become incorporated into the body of the embryo. The zygotes of Amphioxus and amphibians form blastulas of this type. Second, two kinds of cells compose the second type of blastula. One cell type gives rise to the embryo proper and the second type of cells forms auxiliary cells. The latter cells function in implantation, nutrition, respiration and excretion and play no part in forming the body of the embryo. Elasmobranchs and teleost fishes, reptiles, birds and mammals possess this type of blastula. Segmentation starts at about 14 to 15 hours after fertilization as the egg moves down through the fallopian tube. At least 4 cell divisions occur, giving rise to 16 cells, collectively referred to as the morula. From there, the cells that form during cleavage up to the blastula stage are called blastomeres. The first cleavage furrow is vertical, passing through the imaginary axis that runs through the animal and vegetal pole. The two daughter cells which are thus formed are called blastomeres. The second cleavage is also vertical but at right angles to the first one, forming 4 blastomeres of equal size. Subsequent divisions occur one after the other in an orderly fashion. The divisions are rapid and the blastomeres become progressively smaller. During cleavage, the number of cells in the embryo increases but the size of the embryo remains unchanged. The ball of cells is referred to as a blastula. Once cleavage has produced around 100 cells. The blastula is made up of a hollow spherical layer of cells referred to as the blastoderm, which surrounds a yolk of fluid filled space called the blastocele. In most mammals, including humans, the structure formed next is the blastocyst, a mass of inner cells that are distinct from the blastula. Here the cells in the blastula arrange themselves in two layers, the inner cell mass and an outer called the trophoblast. The inner cell mass is also known as the embryoblast. This mass of cells will go on to form the embryo. At this stage of development, the inner cell mass consists of embryonic stem cells that will differentiate into the different cell types needed by the organism. The trophoblast will contribute to the placenta and nourish the embryo. The blastula then develops a pore or a hole at one end called a blastopore. Since glycogen and other nutrients stored in the egg cytoplasm are used in providing energy for cleavage division, the mass of blastula may become less than the mass of the zygote. During blastula stage of development, a significant amount of activity occurs within the early embryo to establish cell polarity, cell specification, axis formation and regulate gene expression. In many animals such as Drosophila and Xenopus, the mid-blastular transition called MBT is a crucial step in development, during which the maternal mRNA is degraded and control over development is passed to the embryo. Many of the interactions between blastomeres are dependent on cadherin expression, particularly E-cadherin in mammals and epicadherin in amphibians. Amphibian and urchin blastulas are hollow balls. Bird and reptiles are flat sheets of cells at this stage. Placental mammal, including human embryos, are special at this stage. We have a blastocyst which differs from a blastula in that only some cells in the internal part will become the adult body. The next stage of embryogenesis is formation of the gastrula. Before gastrula formation can occur, the cells of the trophoblast are differentiated into two layers. One is an outer layer of protoplasm that contains the nuclei called the syncytium, and the other is an inner layer of well-defined cells called the layer of Langerhans. The trophoblast cells do not help to form the embryo proper, but form the ectoderm of the chorion and contribute to the development of the placenta. During gastrulation, the blastula folds upon itself to form the three layers of cells. Each of these layers is called a germ layer, which differentiate into different organ systems. The three germ layers are the endoderm, the ectoderm and the mesoderm. The ectoderm gives rise to the nervous system, skin and hair. The mesoderm gives rise to the muscle cells and connective tissue in the body, and the endoderm gives rise to columnar cells found in the digestive system, lungs, bladder and many internal organs. Now, let us look at the different types of blastula. Types of blastula. Structure of blastula varies in different animals. It depends upon many factors such as size of the egg, amount of yolk in the egg, pattern of distribution of yolk in the egg, and type, rate and number of cleavage division. There are 6 main types of blastula. First, stereoblastula. It consists of large but fewer compactly arranged cells without or with a very small blastocele amongst them. It is formed as a result of spiral cleavage. It is found in annelids, insects and mollusks. Where the blastocele is obliterated by compression of the animal and vegetal poles towards each other, the blastula is termed placula, example Nereis. Second, coeloblastula. It consists of one to many layers of cells arranged as an epithelium around a spacious blastocele. The later may be central or eccentric. Coeloblastula is found in starfish and frog. In starfish, the blastoderm is single layered and blastocele is central. This simple blastula is formed in animals having radial holoblastic cleavage. In it, every cell of the blastoderm is in contact with the hyaline layer externally and in contact with the blastocele internally. The blastoderm is formed of a single layer of cells. Its blastocele is filled with mucopolysaccharide. In microlecithal egg, blastocyst is centric. Third, discoblastula. It is formed as a result of discoidal cleavage. The blastocyst is small and it is called as subgerminal space. It is situated below the blastoderm. Cleave produces a disc of many layers of cells over the top of the yolk. A fluid-filled space called subgerminal cavity appears between the disc and the yolk. Some cells of the blastoderm migrate into the subgerminal cavity. These cells continue to divide and later form a layer over the yolk surface. The new space between this layer and the disc is called the blastocyst. Example: bony fishes, reptile, birds and prototherian mammals. Fourth, superficial blastula. It develops from centrolecithal egg by superficial cleavage in insects. It consists of a single layer epithelium, the blastoderm, enclosing yolk instead of blastocyst. The blastocyst is absent. The yolk contains scattered energids which have not shifted into periplasm. There is a cluster of pole cells at the posterior pole. These cells are formed by cleavage from the pole plasm after it receives some energids. Fifth, amphiblastula. It is formed of two types of structurally different blastomeres. Example: in cycon, the anterior half of blastula is formed of flagellated cells, while the posterior half is formed of large rounded granular cells. In amphibians, the two types of cells are micromeres and macromeres. Sixth, blastocyst. It is formed in mammals as a result of holoblastic cleavage. Outer cells are called as trophoblasts or cells of Robber. They form a trophoderm. This layer gets attached to the uterine wall. Inner cells form the embryo and are called as inner cell mass. Example: human beings. The blastula plays an important role in the development of the individual and represents the first embryonic stage after fertilization. A blastular stage must be formed before the embryo can follow the sequence of steps leading to the establishment of the normal adult animal. The techniques of vital staining or marking of blastomeres enables biologists to construct maps of the prospective areas of the embryo. The labeled cells can be traced as the embryo develops and the fate of various areas followed. The mapping process reveals that the certain cells give rise to certain germ layers, which in turn make no specific tissues and organs within the embryo. They have a predetermined destiny and under normal conditions give rise to specific major organ forming areas of the future adult. These germ layers are ectoderm, mesoderm and endoderm, terms that mean outer, middle and inner skins and refer to their position within the embryo. Since the cells of the blastula are located on or near the surface of the embryo and as yet do not exist in layers, the area of the future germ layers during the blastula stage is referred to as presumptive. For example, that area of blastula destined to form the neural ectoderm is called the presumptive neural ectoderm. The early biologists constructed maps of the blastulas based on the trajectories of tagged cells. They showed the fate of any region in the blastula under normal developmental conditions and were referred to as fate maps. According to these maps, similar patterns exist in all vertebrates. Regardless of the specific shape of the blastula, round or disc shaped, the organ forming areas are arranged in a pattern around the presumptive notochordal region. Clinical significance of blastula. The study of the blastula and of cell specification has many implications on the field of stem cell research as well as the continued improvement of fertility treatments. Embryonic stem cells are a field which, though controversial, has tremendous potential for treating disease. By manipulating the cell signals during the blastula stage of development, various tissues can be formed. This potential can be instrumental in regenerative medicine for disease and injury cases. In vitro fertilization involves implantation of a blastula into a mother's uterus. Blastula cell implantation could potentially serve to eliminate infertility. Blastula stage cells can behave as pluripotent stem cells in many species. Pluripotent stem cells are the starting point to produce organ-specific cells that can potentially aid in repair and prevention of injury and degeneration. Combining the expression of transcription factors and locational positioning of the blastula cells can lead to the development of induced functional organs and tissues. Pluripotent xenopus cells, when used in an in vivo strategy, were able to form into functional retinas. By transplanting them to the eye field on the neural plate and by inducing several misexpressions of transcription factors, the cells were committed to the retinal lineage and could guide vision-based behavior in the xenopus. Summary: the blastula is a hollow sphere of cells referred to as blastomeres surrounding an inner fluid-filled cavity called the blastocell, formed during an early stage of embryonic development in animals. Embryo development begins with a sperm fertilizing an egg to become a zygote, which undergoes many cleavages to develop into a ball of cells called a morula. Only when the blastocell is formed does the early embryo become a blastula. The blastula precedes the formation of the gastrula, in which the germ layers of the embryo form. The 6 main types of blastula, depending upon size of the egg, amount of yolk in the egg, pattern of distribution of yolk in the egg, and type, rate and number of cleavage divisions are stereoblastula, coeloblastula, discoblastula, superficial blastula, amphiblastula and blastocyst. The blastula plays an important role in the development of the individual and represents the first embryonic stage after fertilization. Thank you.